Please stand up, please lead in prayer.
Our Father in heaven, give us this day our daily bread, as we forgive those who sin against us, and lead us not into temptation, but deliver us from evil.
Okay, good afternoon, class.
Good afternoon, sir.
Thank you, be seated. Okay, where is class secretary? This Rice, and can you please report to me the attendance for today?
There are 23 males and 23 females. All are present.
Very good. You give yourselves a round of applause. Do we have assignment? Okay. So today is due day, which means a new opportunity to learn new things. Do you want to learn something new today? Masyadong mahina. Do you want to learn something new today? Okay, but before we proceed with our topic, let us have our classroom groups first. Please read, everyone.
Listen carefully when others are speaking. Be active and share your ideas with others. Respect everybody's thoughts and opinions. Be honest. Raise your hand if you want to answer. Participate and cooperate with your groupmates.
Good. Always remember that's the rules now. So let us start our journey about exploring our topic for today with a game called calculating. Do you want to play a game? Again, do you want to play a game? So I will divide the class into 4 groups. So starting from here, you count 1 to 4. 4, 3, everybody, go to your group mates and form a circle. I will give additional points to those group who can form a circle. Isa mo ka na? 3 and 4. Again, group 1, 2, 3 and 4. Are you ready? I said, are you ready? So what are you going to do? The class will be divided into 4 groups. Each group will be given a manila paper. Attached to the manila paper are the hidden terms that need to be identified by subtraction and addition of words. The first group to present their answers on the board will receive 5 points followed by 3, 2, and 1. Every correct answer is equivalent to 1 point. Group 1, do not open yet. 2, 3, and 4. Again, tanongin ko kayo, are you ready? You may start. You may start now. Again, always remember our classroom rules. Be cooperate with your group mates. Be active and share your ideas with others. Respect others' opinions and thoughts. The first group to present their answers on the board will receive 5 points, followed by 3, 2, and 1. Every correct answer is equivalent to 1 Tapos na ba? Malapit na. Teamwork, teamwork, teamwork. The team that works together wins together. Last minute. Thirty seconds. If you want to place your answers on the boards, the tapes or discussion sheets are available here. Everyone, you. Three, 10 seconds. Group 1 and 2. 5, 4, 3, 2, 1. Finish or unfinish, post your answers on the board. Okay, let us check your work. First, Chandra minus RE plus ethics minus HS is equal to? Very good. Next, engine plus ear minus A plus side range plus minus sire is equivalent to? Engineering. Very good. Does everybody got the correct answer? Geometry and engineering. Very good. Retry minus try plus combination minus Sean plus ant minus A is equal to? Recombinant. Very good. So far, lahat ay nakatama. Next. Tide minus tide plus die minus die plus C minus SE is equal to? Very good. DNA. DNA. Ano sa group 2? Sorry to say, but this is not DNA. It's DINA. So group 4 got the correct and correct answer for item number 4. Let us proceed with the last word. Teach minus A plus novel minus VE plus logo minus O plus Y is equal to? Technology. Very good. And shall we give yourselves a nice clap? Do you know what is a nice clap? N-I-C-E. N-I-C-E. Nice, 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 nice. Everybody, 3, 2, 1, go.
N-I-C-E. N-I-C-E. Nice, nice.
For every correct item is equivalent to 1 point. For group 1, how many points do you get? 5. For group 2, 4. For group 3, 5. For group 4, 5. For our next activity, I want you to look at this picture. What can we see? Picture.
Genetic.
What?
Genetic.
Genetic? Okay, you have your point. Genetic.
DNA.
Very good. It's DNA. What have you noticed with the DNA? Anyone? Yes, Mr. What is your family name? Dalman. Yes, Mr. Dalman. What can you see or what have you observed with the DNA in the picture? Ano tawag dito? May nakuha ba? Yes, it's very good. DNA is the DNA, something that's been removed in a DNA molecule. Now, for picture number 2, what have you noticed? What can you see? Oh, very good. What have you noticed with the sizes of the forms? Yes, you may stand, Mr. Sardilio. Yes. Then what have you noticed with the sizes? The other one is big and the other is small. So there's the difference between the sizes of the form, which means that something has changed. Based on these features and the words you have identified earlier, what do you think would be our topic for today? Anyone? Yes, Miss Apeliedo. Raise your hand kung sa daw. Apeliedo. Avila. Yes, Miss Avila, what do you think would be our topic for today? Na-identify niyo na kanina, 5 words plus yung additional na features. Yes? Our topic for today is about genetic or kulang pa. Anong genetic? Yes! Si Miss ano daw? Ano gusto mo? Gabrielo? Gabrielio. What do you think would be our topic for today? Based on these features and the words you have identified earlier, it is all about genetic, malapit na, organ systems. Klaro na yung kanina, nandito yung isa, dito yung pangalawa. You raise your hand if you want to answer. Yes, Apeliedo. Santillan. What is our topic for today, Ma'am Santillan? Very good. Let us give her a 5 claps. 1, 2, 3, 4, 5. Our topic for today would be recombinant DNA technology or genetic engineering. At this point, let us read our learning objectives. Everybody, in 3, 2, 1, go.
At the end of the lesson, students will be able to define recombinant DNA technology or genetic engineering, explain how recombinant DNA is created, and recognize the importance of genetic engineering.
So, to start our exploration as to what is genetic engineering, let us have another game. Do you want to play another game?
Yes!
Masyadong mahina. Do you want to play another game?
Yes!
Dapat, sir, yes, sir. Do you want to play another game?
Sir, yes, sir!
Very good. This game is called Find My Place. Tanong niyo sa katabi niyo, may lugar ba ako dyan sa puso mo? What are you going to do? With the same group, each will be given an envelope containing terms or words and an empty concept map related to genetic engineering. We are going to complete the concept map using those terms. You will be given 3 minutes to finish the task. Every correct answer is equal to 1 point. For group 1, Group 2, Group 3, and Group 4. Do not open yet. Are you ready? Are you sure? Are you ready? Oh, nakalimutan na agad. Are you ready?
Yes, sir!
Very good. So your timer starts now. May double-sided sa likod. As you may have noticed, may mga boxes dyan na hindi kasya yung terms kasi masyadong mataas. All you have to do is pwede niyong i-fold, basta lang nakalagay siya sa tamang lalagyan. Para mas madali yung iba tagatanggal ng double-sided tape sa likod, while the others are thinking as to where it will will be put. Always remember as rules, cooperate and participate with your group mates. Be active and share your ideas. Respect everybody's thoughts and opinion. Again, ang instruction, no, as you may have noticed, may mga boxes na hindi kasya yung mga words kasi masyadong malaki or mataas. All you have to do is to fold. Pwede nyo i-fold as long as it is put in the right box. You have 90 seconds left. Another instruction, as you may have noticed, masyadong malaki yung concept map when being put in a drawer. You ask your group mates to somebody from your group to hold the concept map during our checking of your answers. You have 50 seconds left. Seconds left. Ten, nine, eight, seven, six, five, four, three, two, one. Finished or unfinished, you raise your answers. Again, I will deduct the points of those groups who will not follow instruction. We ask one or somebody from your group to hold your answers. Yarasa, you stay with your group mates. You stay with your group mates. Yep. Diyan ka ate sa Eagle. Somebody, may take your seat, except for those who hold the bagsak. Are you confident with your answers?
Yes.
Why such? Are you ready to check? Folks, are you ready to check?
Yes.
You raise? You raise? Oh, no more? Let us check your answer. Recombinant, again. You raise? Kuya, you raise your answer. The others, you may take your seat. Ask just somebody to raise your answer. The others will have to take their seats. Again, to check your answer, recombinant DNA technology or genetic engineering. There are tools in genetic engineering. Una, enzymes. Does everybody got the correct answer?
Yes.
Oh, endonuclease. Endonuclease, polymerases. Tama ang enzymes pero nakalagay sa maling box. Dito, walang nakatama. Second tool of genetic engineering, vectors. Very good. Group 1, you get the correct answer, 1 point. Group 2, mali, mali, at saka mali. Another tool, or the last tool, host organism. Mali, 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 mali. You remember your point, 1 point. Now, enzymes are the type of tools in genetic engineering is enzymes. There are types of enzymes. What are those? Restriction enzymes, polymerases, and ligases. Let me check. 1, 2, very good. Mali. 3 points. 1, correct. 1, 1 point. 1 point, 1.3. Mali. Now these restriction enzymes are subdivided again into 2. What are those? Endonucleases and exonucleases. Let me check. Mali, mali. May tamang isa, 2 points na kayo. Very good. 1 and 2, 3 points na kayo. Dito. Oh no. Exo and endo. Very good. 2 points. Ilang points na kayo? 3. Then from sa last, vectors. Most common vectors. Plasmids and bacteriophages. Let us check. Tama ang isa. Ilang points yun na? 4. Mali. Mali. Okay, very good. 1 plus 2, ilan na kayo? 3. 5 na. 3 kayo kanina. Okay. You may take your seat. I know that it's good that everybody has or might identify some of the terms. And I know some of you are still confused as to what is the interconnection between these terms. Anong si please naman gibutan? Anong si restriction enzymes they are man? Let us find out as we dig deeper into these terms through another game. Do you want to play another game? Yes. Boy, kalimutan niyo na. Do you want to play another game? Sir? Yes, sir. Again, they want to play another game? Yes, sir. Let's play a game called Find My Match. Nakita niyo ba na ba ninyong ka-match? Wala pa. Studies. What are you going to do? With the same group, each will be given a Manila paper. Attached to the Manila paper are aforementioned terms earlier along with its corresponding information. You are going to match the terms to its corresponding information. You will be given 3 minutes to finish the task. The first group to present their answers on the board will receive 5 points, followed by 3, 2, and 1. Every correct answer is equivalent to 1 point. Are you ready? Sir. For group 1. 2, 3, wait. 4. So, mauna siyang inyong terms and the information na inyong i-match. Your timer starts now. 20 seconds. Double time, everyone. One minute left. Again, please be reminded by our lesson rules. Cooperate with your roommates. Share your ideas and respect everybody's thoughts and opinions. 20 seconds left. 10, 9, 8, 5, 4, 3, 2, 1. Okay, time is up. Markers up. Again, time is up. Markers up. I will deduct points to those who did not follow instruction. Again, markers up. Markers up. You paste your answers on the board. Group 1, followed by 2, 3, and 4. This time, I will ask you again, are you confident with your answers? You're confident? Let us check your answers. So, let us check your answers and at the same time, let us dig deeper into these words. First, with our main topic, recombinant DNA technology or genetic engineering. Ano ba to? Genetic engineering involves the group of techniques used to cut up and join DNA, especially DNA from different biological species, and to introduce the resulting hybrid DNA into organisms in order to form new combination of heritable genetic material. Let us check. Group 1 got the correct answer. Group 2 and Group 3. Kung atong tinambal ng pag-define kung saan yung recombinant. Okay, my bad. Group 2 got the incorrect answer. Okay, so kung saan man ito pag tinambal ng pag-define ng recombinant DNA technology or genetic engineering. From the word recombinant, asa man naging na word? It's from recombination or recombining. Kung saan man yung recombine? DNA molecule. Also may connection ni technology sa recombinant o ang DNA. When we talk about technology, it is directly associated with manipulation of gadgets. Am I right? Yes, sir. So kung atong buhol, recombinant DNA technology, it is about recombination of DNA molecules through manipulation with the aid of technology or technological devices. The same goes with genetic engineering. When we talk about genetic, of course, genes, engineering. Kung sa may direct or association ng engineering there. When we talk about engineering, it is directly associated to the word construction or reconstruction. When you were young, you dreamt of becoming an engineer to build your dream home or desired home. So kung atong tinambalan o buuhon, basta din ang genetic engineering. It is about construction or reconstruction of DNA molecules containing your desired genes. So kaning genetic engineering has your different tools na gamit. What are those? First is the So what is or what are enzymes? Enzymes are proteins that help speed up metabolisms or the chemical reactions in our body. Let us check. Asa man si enzymes? Mali. Si enzymes, mali. Si enzymes, mali. Saka si enzymes. Very good. Si group 4, nakatama. So, enzymes are proteins that help speed up metabolism or chemical reactions in our bodies. These enzymes are subdivided into three. What are those? Everybody, what are those types of enzymes? So what is restriction enzymes? Restriction enzymes are proteins that recognizes a specific short nucleotide sequence and cuts the DNA only at that specific site, which is known as the restriction site or the target sequence. Or simply say, restriction enzymes help to cut. They play a major role in determining the location at which the desired gene is inserted into the vector genome. So what's the restriction enzymes? It help to, let us check. Restriction enzyme, mali. Restriction enzyme, mali. Restriction enzyme, mali. Then restriction enzyme, mali rin. So walang nakakuha sa restriction enzymes. Now, when we talk about restriction enzyme, may dalawang pwedeng forms. Because we're talking about cutting. So, ang isa, ano ang isa? Endonucleases. The other one, exonucleases. Let us first talk about endonucleases. When we talk about endo, what does it mean? Endo. Nasa sulod, nasa gawas. Very good. Endonuclease is a type of restriction enzyme that cleave to divide or split the nucleic acid strand at the middle, inside or within from that it might be formed sticky or landed. So, ano ito kung ingon? Si restriction enzyme may kabuok, si endo o si exo. Karon, si endonuclease kay tungaon man sa tunga, nakapwede ang form. It's either sticky or landed. But let us first check kung nakatama ba kayo sa endonuclease. Very good. Middle. Mali. Mali. At saka mali. So, what's the meaning sticky o blunt ends? When we say sticky end, one strand is longer than the other such that the longer strand has bases which are left unpaired. What's the mga bases? Asa may bases nila, babe? Kani. C-A-T-C-G. When you were younger, when in your lower years, you were already taught about the base pairing when it comes to DNA sequence. What are those base pairing? A, T, C, G. Adenine paired with? Masa may pair ni adenine? Cytine. Masa ba na cytine? It's thymine. Cytosine is paired with? Masa ba na si G? Guanine. So, always remember that because when you're going to college or any other, when you're going to discuss DNA, the base pairing will be ATCG. It would be adenine paired with thymine, cytosine paired with guanine. So, sisnikin, the one strand is longer than the other. One strand is longer than the other such that the longer strand has bases which are left unpaired. O sa mga po ng kalayan niya sa isa pa ka-N. C, lang N. Both strands are of equal length. When we say length, unsa na siya? Very good, ang katasons. As you can see, balance kayo ang pagkakag nila. So, unsay kaliyan ana? Ang isa, longer ni sa strand kontra sa isa, tapos 9 bases nga left and paired. Pag blunt end, both strands are cut with equal length. So karon sa restriction enzyme umanta sa endonuclease. O sa na po ng exonuclease? When we say exonuclease, it is a type of restriction enzyme that cleave the nucleic acid strands from end. When we say exo, sulod gawas. Gawas or from the end. So, kita kayo dira sa picture ang activity ninyo sa kung sa itong pagkakat sa endonuclease, asa na ba? Sa middle, within, or sa tuma. Si exo sa end or sa gawas. We have done already talking about the first type of enzyme, restriction enzyme. Let us proceed with the other type of enzyme, the polymerases. What is DNA polymerase? Everybody read.
It's a type of enzyme that is responsible for the process of DNA replication.
Very good. DNA polymerase is a type of enzyme that is responsible for the process of DNA replication. Kung saan ang DNA replication? When we say replication, kung saan na siya? Forming new copies of DNA. Identical, unidentical? Identical, very good. When we say DNA replication, it is about replicating or forming new copies of DNA that are identical to each other. So simply put, DNA polymerase helps to synthesize. Let us check your answer. Very good. Tama. Mali. At saka mali. Now, anong last type of enzyme? It's the, everybody read. How do you read that one? Ligases. Everybody, ligases. So what is an ligase? DNA ligase is a specific type of enzyme that facilitates the joining of DNA strands together. Or simply put, DNA ligase helps to kung saan man ang bind sa Bisaya pa. Very good. It's to bind, join, or ano bang tawag dyan? To connect. So tapos na tayo sa first tool of genetic engineering, the enzymes. Let us proceed with the second tool. What is the second tool in genetic engineering? Vectors. So unsa man ang vectors? Vectors is the ultimate vehicles that carry forward the desired genes into the host organisms. Later on, makibatan ninyo ang purpose ni vector. You're going to use vectors to insert your desired gene before introducing it to the host organism. So there are most common type of vectors. What are those? The two most common vectors are plasmids and bacteriophages. Everyone, bacteriophages. So let us first identify what is a plasmid. Plasmid is a small circular DNA molecule found in bacteria and some eukaryotes. It is an extra chromosomal DNA molecule within a cell that is physically separated from the chromosomal DNA and can replicate independently. So nakita ninyo diyan sa picture. So nga sa picture? Bacterial cell. So natin si bacterial DNA, natin si karon. Kung saan mo ng other type of most common vector? It's the again, kung saan na? Everybody. Bacteriophages. Next. What is bacteriophages? Bacteriophages, also known as phages, are viruses that infect and replicate only in katong galing yung ipakita. Kung saan ito tawag ito? Bacterial cell. Very good. So let us check your answers. Vectors. Kung saan ganitong vectors? Is the ultimate vehicle to carry your desired genes. Vectors. Wrong. Very good. Vectors. Mali. Vectors. Hindi klaro pero sila cases klaro-klaro. So, it's incorrect. So, the group 1, group 2 got the correct answer. What are the two most common vectors? Plasmids and bacteriophages. So, unsa man si plasmids?
A small circular DNA.
So, let's check. Mali. Mali. Plasmids. Mali. Si plasmids. Tama. What is the other common vector? What is a bacteriophages? Are viruses that infect and replicate only in
Bacterial cells.
So, nasa na si bacteriophages? Mali. Mali. Very good. Group 2. Group 1. Mali. So now we're done discussing the second tool in genetic engineering, which is the vectors. Now, what is the last tool in genetic engineering? Okay? Very good. It's the host organism. What is an host organism? Everybody read. Okay, so muna tayo sa first word. Host organism is where recombinant DNA is.
Introduced.
Let us check. Tama ba ang lahat? Host organism?
Tama.
Mali. Mali. Group 3? Mali. Group 4?
Tama.
Very good. There, recombinant DNA is introduced. Now, these host organisms are there are various ways in which these recombinant DNAs are inserted into the host, namely alternate heating and cooling, use of calcium ions, microinjection, bioelastic, or gene. Sorry na microinjection. Microinjection is a technique wherein substances are injected into single cells using a very thin needle. As you can see in the picture, anong tawag dito? Injection. Manipis or masyadong malaking needle? Manipis. Anong tawag dito? Host cell or the cell, single cell where the substances is to be injected. Ngayon, ano naman, may other way pa ng pag-introduce o pag-insert ng DNAs into the host organism. The other one is the bioelastic or gene gun. What is bioelastic or gene gun? Everybody read. As you can see in the picture, physical introduction of DNA into the plant cell. So those are terms, or those are the interconnection of these terms related to genetic engineering. Now, let me ask you, when we talk about genetic engineering, do we talk of it as a process? Anyone? When we talk about genetic engineering, do we talk of it as a process? Yes. Why do you say so? When we talk about genetic engineering, we talk of it as a process. And when we talk about the process, there are steps to be followed because it should be done systematically. When we say systematically, it is a step-by-step process. The same goes with genetic engineering. There are steps to be followed. What are those steps? Let us find out through another game. This game is called Arrange Me. Do you want to play another game?
Yes, sir.
Masyadong mahina. Do you want to play another game? Very good. What are you going to do? With the same group, each will be given another envelope. Inside the envelope are the steps in recombinant DNA technology or genetic engineering. We are going to arrange the correct sequence of the steps. We will be given another 3 minutes to finish the test. The first group to base their answers on the board will receive 5 points, followed by 3, 2, and 1. Every correct answer is equal to 1 point. Before we do not open yet, before we proceed, let us record your scores first. Ilan na po ang points ng group 1? 4. For group 2? Ang group 3? Ang group 4? Okay, are you ready? It's for group 1. Sa likod. Are you ready? So, your timer starts now. The first group to present their answers on the board will receive 5 points, followed by 3, 2, and 1. Okay, the first group to present their answer is the group 1, followed by group 2. You still have a minute. Okay, those who are finished may take their seats. Okay. Sure na ba kayo sa answer ninyo? Are you sure? Let us find out and let us check your work. The steps in genetic engineering or recombinant DNA technology starts with the selection and isolation of DNA insert. Everybody read the first step. Again.
First.
3, 2, 1, go! So the first step is the selection and isolation of DNA insert. As you may have noticed here in our example, it is summarized process using the bacterial plasmid as a vector. So, asa niya natin itong itawag ng selection and isolation of DNA insert? So first thing, they have chosen their DNA. What type of DNA? Human DNA containing the desired gene. What is the desired gene? Very good. That's the first step, selection and isolation of DNA insert. Next, who got the correct answer? It's the group 4. So you got 1. Next step. Everybody read. Again. 3, 2, 1, go. Step is the selection of suitable cloning vector. Based on this example, anong ginamit nilang cloning vector? Based on this feature, anong ginamit nilang cloning vector? Again, the most common vectors are plasmids and bacteriophages. So based on this feature, or in this process, anong ginamit nilang vector? Very good. It's the bacterial plasmid. Next, what is the third step? So before that, let us check the answer. So the third step is the, everybody read. The third step in genetic engineering is to introduce the DNA insert or the clone DNA into the vector to form our DNA molecule. In this example, saan part dyan sa introduction of DNA insert? First, you're going to the selection of your desired DNA containing your desired gene. Human DNA containing the desired gene protein, the gene. The next step would be what? The selection of suitable cloning vector. So after that, you're going to cut the DNA molecule into thousands of fragments. Ayan na. Thousands of fragments yan. Tapos nandito na si desired gene. Before that, you're going to cut the bacterial plasmids into the, anong tawag doon sa specific site? It's the target sequence. So ayan na. Based on this feature, anong klaseng pagkakat ito? Ano bang ginagamit pagka? Anong enzyme? We have already discussed that earlier. It's the restriction enzymes. So nakat na siya. Anong klaseng pagkakat to? Indo or exo? Very good. It's endonuclease. What type of end ang na-form? Sticky or bland? Very good. Sticky. Ngayon, nandiyan na si desired DNA, si cloning vector. What we're going to do is to introduce. Ayan na. Kinonect na si DNA insert or the clone DNA to the vector, which is the... Ngayon, pag nag-join na yan, anong gagamitin pag nag-join? Anong enzyme? It's the polymerase or ligase? Very good. It's the ligase. Now, the next step is the... Everybody read.
This process of entry of our DNA into the host cell is called transformation. Usually selected hosts are bacterial cells like yeast, however, yeast and fungi may also be present.
Everybody read. How do you read this one? E. coli. Everybody, again? Again. So the next step is the recombinant DNA molecule is introduced into a suitable host. So ayan na kanina. Si vector na bacterioplasmids contains the desired gene, which is the protein B. Connected na sila or joined by what type of enzyme? Ligase. Ngayon naman is to introduce the recombinant DNA into the suitable host. Anong host ang pinili nila dito? What? Very good. Under the right conditions, a bacterial cell takes up the recombinant plasmid. So meaning to say, the bacterial cell is the suitable host. Next. Step number 5. Everybody read. Selection of transform host cells. Ano nga ba daw transform cells? Recombinant cells are those host cells which have taken up the rDNA molecule. In this example, nasaan si rDNA molecule? It's the cloning vector containing the desired gene. Ngayon, sa step 5, you're going to separate the non-transformed cells, meaning to say, those who do not have taken up the desired gene will be separated to those who have taken out. The last step in genetic engineering is the expression and multiplication of DNA inserts in the host. Everybody read. Finally,
it is to be ensured that foreign DNA inserted into the vector DNA is expressing the desired character in the host cell. Also, the transformed host cells are multiplied to obtain sufficient number of copies. If needed, such genes may also be transferred and expressed into another organism.
Finally, the last step in recombinant DNA technology is the expression and multiplication of DNA insert, or clone DNA are the same in the host. Finally, it is to be ensured that the foreign DNA is inserted into the vector DNA. Kung sa may pangalan, asa man diri si foreign DNA. It's the desired gene which is the protein B gene. So ensure daw niya nakain na sa vector. What type of vector? Bacterial plasmid. Ngayon, also the transformed host cells, this is what you call the transformed host cell, are multiplied to obtain sufficient number of copies. If needed, such genes may also be transferred and expressed into sufficient. Multiplied to obtain sufficient number of copies through what? So, i-replicate na niya tong host cells na naana si recombinant DNA molecule. So, sama na niya through DNA replication. Very good. What do you mean by DNA replication again? It is to form new copies of identical DNA. Through what? Sa usang pamagi man ng DNA replication. Through cell division results in many identical copies of the bacterial cell, all carrying the protein B gene, which is the desired gene. When the genes are expressed, multiple copies of protein B gene are made. So, ano yung mga copies tayo? So, those are the steps in genetic engineering. Now, is genetic engineering important?
Yes, sir.
Yes, sir. Why do you say so? Sige nga, kuya. Apelyido. Daga. Yes, Mr. Danga. Why do you say so that Daga? My bad. So, what do you say so that genetic engineering is important? Marami bang gamit si genetic engineering? Marami, wala? Marami, yes, very good. Genetic engineering is important because it has a lot of applications. Some of those are the following. Everybody read the first application. By utilizing the tools and techniques in genetic engineering, it is possible to produce transgenic plants. What do you mean by transgenic plants? Transgenic plants are genetically modified plants. Are these plants useful and beneficial? Yes, it is, because it increases the crop yields, it reduces the cost of food and crop production, it reduces the need for kasi ginagamit na lang sa mga farmers para pampawala sa mga pests. Yes, very good. Is the pesticide that it enhances nutrient composition and food quality. It enables resistance to diseases or some pests, and most importantly, it provides greater food security. The next application would be, everybody read. So by the use of RDNA technology, desired genes can be inserted into animals so as to produce transgenic animals, or also known as genetically modified animals. So important ba ng genetically modified animals? Yes, because this leads the animal breeders to increase the speed and range of animal production or farm animals that are resistant to diseases. What are example of transgenic animals? Transgenic cattle. Kaino ba mo si cattle? Wala. Baka. When you refer to, what is the difference between cows and cattle? When we say cattle, it is used when we say general. It does not depend on the gender. Kahit palalaki o babae, it should be called cows. When you refer to cows, it should be referred to cattle, my friend. Pag sinabi natin cows naman, kasi yan yung misconception nila. When you say cows, it refers to a mature female cattle. So dapat, it's more appropriate to use cattle than cows. So ano bang gamit ni transgenic cows? Transgenic cattle were created to produce milk that contains specific human proteins in order for the treatment of human emphysema. Kung saan man emphysema, sir? Kung saan man emphysema daw? Emphysema is a lung disease that causes the shortness of breath. So gamit na gamit talaga si genetic engineering in production of transgenic animals. Next, the third application. Again? What is a vaccine? A vaccine is a chemical preparation containing a pathogen in an active state that can be introduced into humans or animals to confer immunity against infection. Kung may examples ng mga vaccine, sirs? Na-vaccine nga bang laban against polio, malaria, cholera, hepatitis, smallpox, and karon, sa ginamit na vaccine against COVID-19. Next application. So, man daw ng antibiotics. Antibiotics are medications that destroy or slow down the growth of bacteria. Kung saan gamit diha may genetic engineering. Genetic engineering helps increase the production of antibiotics by improving the microbial strains through modification of genetic characteristics. So, may most common example of antibiotic. Yes, dapat na siya. Antimicrobial. Most common is the very good. It's the penicillin. The next application. Prevention and diagnosis of diseases. We can use genetic engineering to prevent diseases through fixation of mutated genes. Katong mga genes na naka-cause of diseases, pwede siyang tanggalon. Tapos katong mga genes na maka-help paglaban sa diseases, mas padaghanon. Next is the gene therapy. What do you mean by gene therapy? Gene therapy is the treatment of diseases by transferring an expression of genes into a patient's cells in order for it to restore a normal cellular activity. Gene therapy is the most beneficial area of genetic engineering. Lastly, applications in forensic science. Actually, this is the most common. Nakita na ninyo dito sa, kasi nakakita sa link ng or series, Detective Conan, sa Nabus ka ng sa Sopo, sa ba? Si 007 James Bond. So, genetic engineering is very applicable in forensic science. The application of RDNA technology in forensic science largely depend on the technique called RDNA or DNA fingerprinting or DNA profiling. Saan yung gamit na sir? It is to identify person using his or her hair roots, food stain, serum, and etc. Paggamit kayo ng genetic engineering and forensic science to identify those kung sa man ang tawag ano nakasala, nakakomit of crime. It's green. Very good. It is helpful in identifying criminals. These applications are just some of the applications of genetic engineering. Daghan na sila kaayo. Karon, that would be all for the applications and steps and about genetic engineering. Do you have any questions? Wala na. So to test your understanding, let us have a short quiz. But before that, please go back to your original seats and prepare a pen. In 10, 9, 8, 7, 6, 5, 4, 3, 2, 1. Everybody raise your chairs and prepare a pen. Keep all the unnecessary things put in on your bags, including your books.
Cut na sir, cut na.